Hello, boys and girls. In today's activity, we're going to be looking at Activity 4, the history of the cell theory. During this reading, you will need to fill out a student sheet 4.1, which looks like this. It says we go through the different scientists, such as Hooke, Leeuwenhoek, Schleiden, Schwann, and the other guys listed on here. You will need to fill out what their contribution to the cell theory is. If both boxes are open, then they contributed to both the cell theory and the germ theory of disease. Schleiden and Schwann, their only contributions was to the cell theory. Therefore, that's why these blocks are grayed out. When you get down to J. Robin Warren and Barry Marshall, their contribution is just to the germ theory of disease. So you won't write anything here. That's why these boxes are grayed out. So just want to make sure that you are aware on how that worked. Okay? So during the video, as you come across these contributions and you write things down in these boxes, just pause it. Pause the video. Okay? All right, so here we go. The history of the cell theory. Scientific discoveries often follow the development of new tools and technologies. This is certainly true in the case of infectious diseases. In the 1890s, researchers Alexander Yersin and Shabrasabro Kazatso independently used a microscope to identify the cause of the bubonic plague. By the way, I think that's how you pronounce them. Terrible with pronouncing names, so I apologize. Compound microscopes that use more than one lens, had been invented around 1595. The first microscopes usually magnified objects only 20 to 30 times their original size. But as you will learn, even this level of magnification was enough to discover a world of new scientific ideas. By 1840, Italian physicist Giovanni Amici had invented the oil immersion microscope, which could magnify objects 6,000 times. In most middle school, the highest level of microscope magnification is usually about 400 times. Today, the transmission electron and the scanning electron microscopes can magnify objects over 40,000 times. All right, so the guiding question is, how did the cell theory lead to the germ theory of disease? The materials you have in front of you should be student sheet 4.1, which I pointed out to you earlier. Okay. All right, so here we go with the reading. Robert Hooke, 1635 to 1703. Late 17th century was a period of great scientific discovery. While many people offered theories without experimentation or evidence, English scientist Robert Hooke believed that good science resulted from making observations on what you could see. In his 20s, he wrote a book of his observations and drawings of the natural world called Micrographia, meaning tiny drawings. It was first published in 1665. In this one book, he presented the ideas about the life cycle of mosquitoes, the origin of craters on the moon, and fossils. But Hooke is most remembered for including drawings of what he saw through a microscope. Hooke developed his own version of the compound microscope, and it was one of the best available at the time. Today, his most famous drawing from Micrographia is a drawing of cork, the same kind of cork that is used in cork boards and bottle stoppers. Since cork is made of the bark of the cork oak tree, it is essentially dead plant tissue. Using his microscope, Hooke looked at very thin slices of cork. He noticed what looked like little rooms below on the right. Because of this, he called these shapes cells, another word for rooms. With a simple observation, Hooke introduced an idea that would become the basis of new fields of biology, but not for almost 200 years. There's a cork tree, and there is what cork cells look like under the microscope. Antoine van Leeuwenhoek, 1632 to 1723. Antoine van Leeuwenhoek was a cloth salesman in Holland and an amateur scientist. He knew how to make very simple microscopes. Today they would be considered magnifying glasses. But he did not become interested in studying the microscopic world until he read Hooke's Micrographia, which was a very popular book at the time. Leeuwenhoek still or skill at building microscopes, like the one shown at the right, enabled him to magnify objects over 200 times. In 1673, Leeuwenhoek described what he saw in a drop of water. He described them as wretched beasties. They stop, they stand still, and they turn themselves around. They are no bigger than a fine grain of sand. By examining scrapings from his teeth, he found additional evidence of these many very little living animalcules, very prettily and moving. Leeuwenhoek was one of the first people to observe and record microscopic living things. He continued his observations until the end of his life. Again, till this point, no one knew that these creatures existed. No one knew the microscopic world. These gentlemen led to the discovery of that world. Matthias Jacob Schleiden, 1804 to 1881. Theodor Schwann, 1810 to 1882. And Carl Theodor Ernst von Siebold, 1804 to 1885. So let's see what these three gentlemen did. Over the next 150 years, scientists continued to use the microscope to study living organisms such as plants, insects, and microbes. But by the early 1800s, most botanists, scientists who study plants, were not using microscopes. They were busy naming and describing entire plants. German biologist Matthias Schleiden was an exception. Although he trained as a lawyer, he left the law to become a professor of botany. Schleiden preferred to use microscopes to study plants. Look at the plant cells on the next page to see what Schleiden may have seen. We'll look at it in a minute. Based on his study, he suggested in 1838 that all plants are made of cells. This was a completely new idea. Just as a house could be made entirely of bricks, plants were made entirely of cells. Schleiden knew another German biology professor, Theodor Schwann, who spent his entire time studying animals. Schwann was particularly interested in the digestive system. In 1839, 1 year after Schleiden proposed this theory, Schwann suggested that animals, and not just plants, were made of cells. You can see animal cells in the photo below. Because of their ideas, Schleiden and Schwann are credited with developing the cell theory, the theory that all living organisms are made of cells. Other scientists began to build on Schleiden and Schwann's ideas. In 1845, Carl Theodor Ernst von Siebold suggested that microbes were also made of cells, or more specifically, one cell. See the microbial cell in the photo below. Siebold believed that organisms made of many cells, like animals, were built out of single-celled microbes. While Siebold was wrong about that idea, he is right in stating that microbes were living creatures made up of the same material as plants and animals, or animals and plants. Rudolf Karl Virchow, 1821 to 1902. Why is Schleiden and Schwann's cell theory important for understanding infectious diseases? Their work influenced Rudolf Karl Virchow, a Polish doctor. He had been treating and studying ill patients for many years. He is famous for saying in the 1850s, all cells arise from cells, meaning that cells reproduce to create new cells. He was right. When you see a new plant or baby animal, you see a multicellular creature. All living organisms begin as a single cell. Most microbes are made up of only a single cell, as Siebold believed. See the section on what we just read. Single-cell organisms, such as an amoeba, are known as unicellular. So you get to see amoebas when we look at them under the microscope. The cells of multicellular organisms, like people, continue to divide and grow. An adult human being is made up of about 10 trillion cells. All right, so here we go. Plants are made of cells, like the plant cells shown in the photo above. Animals are made of cells, like the skin cells in the photo above. Many microbes are made up of one cell, like the microbes shown in the photo above. Okay, so one thing I want you to notice here, and we'll get to this in a different activity. Notice the shape. Plant cells look like bricks, look like brick walls. By the way, notice these little dots. Make note of that because we'll get back to what that is when we look at plant cells under the microscopes ourselves. Notice the shape, okay? Do you see a difference there? Pay attention to that, okay? That's important, distinguishing between plant cells and animal cells, okay? We'll talk more about it later. Each of these multicellular organisms is made up of millions, trillions of cells. So human being, pine tree, cat. Virchow applied his ideas to disease. He knew that all cells grew from other cells. He thought that all diseases were caused by cells that did not work properly. He believed that diseased cells came from other, usually healthy cells of the sick person. Virchow's ideas about disease were not completely correct. Although they are correct for some diseases, his ideas were based on his work with leukemia, which is cancer of the blood. Cancer and certain other diseases are caused by cells that no longer function as they should. In the case of cancer, the cells tend to grow without control. Infectious diseases are different, as scientists after Virchow discovered. All right, Louis Pasteur, 1822 to 1895. Louis Pasteur, a French chemist, began studying microbes in 1864. He was working on an important business in France, the fermentation of wine and vinegar. He noticed that certain microbes could cause food and drink to spoil. Pasteur discovered that different microbes cause different kinds of spoiling, but heat can kill many of these microbes. Today, because of his work, milk is heated to 71 degrees Celsius for 15 seconds to kill the microbe that causes tuberculosis. Using heat to kill microbes is known as pasteurization, in Pasteur's honor. Look at the milk carton shown at the right. The word pasteurized on milk sold in stores tell you that milk is safe to drink. So again, pasteurization kills microbes that may be present in milk. In 1865, Pasteur was asked to help the silk industry of France, which was having problems with silk production. Silk is produced from threads spun by a worm known as the silkworm. Pasteur observed a microbe that was infecting the silkworms and leaves that they ate. When he recommended that the infected worms and their food be destroyed, the silk industry was saved. Pasteur knew that some diseases were infectious. He suggested that microbes, which he referred to as germs, cause infectious diseases and were easily spread by people. This idea was the basis of the germ theory of disease. Robert Hooke, 1843 to 1910. Slowly, the role of microbes in causing infectious diseases began to be accepted, but there was still more work to be done. Which microbes cause which diseases? In 1876, Robert Hooke, a German doctor, identified the microbe that causes anthrax, an infectious disease that was killing cattle. He later went on to identify the microbes that cause tuberculosis and cholera. Amazingly, he did all of his work in a 4-room apartment that he shared with his wife. Hooke developed a way to prove that a specific microbe caused a particular disease. In the case of anthrax, he injected healthy mice with blood taken from farm animals that had died of anthrax. So here you go. Here's Hooke's experiment. So healthy mice infected with blood from cow with anthrax. Mice die. Anthrax microbes can be isolated from blood of infected mice. So healthy mice injected with healthy cow blood. Mice live. No anthrax microbes in the blood of healthy mice. So back to the sentence here. In the case of anthrax, he injected healthy mice with blood taken from the farm animals that had died of anthrax. He injected another group of healthy mice with blood taken from healthy farm animals. All the mice injected with the blood from one infected animal died of anthrax. None of the other group of mice developed anthrax. He then showed that he could isolate anthrax microbes only from the mice that were injected with blood from the infected animals. He did not find anthrax microbes in the healthy mice. In this way, Hooke was able to provide scientific evidence that a specific anthrax microbe caused anthrax. Hooke also created new ways to show cultures of uncontaminated microbes. In particular, he developed agar, a gelatin-like substance used to grow microbe cultures on a solid surface. Agar is still used today. Substance inside this dish is known as agar. Many kinds of microbes can grow on agar that has added nutrients which serves as food for microbes. There you go. J. Robin Warren, 1937, and Barry Marshall, 1951. The dots there, or the slashes, mean, I don't know if you noticed or not, but this is, when they were born and when they passed away. And I'm assuming that these gentlemen are still alive. Okay? By 1970s, scientists knew that diseases were caused by microbes. They knew other diseases, like heart disease, were caused by unhealthy eating, stress, not enough exercise, and genetic factors. For a long time, scientists thought ulcers were caused by certain foods and stress. They did not think that microbes could cause ulcers because they thought the stomach was too acidic a place for microbes to live. Thought it was too acidic for them to live in that space. In 1982, J. Robin Warren, an Australian, made an important discovery. Warren was a pathologist, a doctor who looks at tissues under a microscope to diagnose disease. When looking at samples of people's stomachs, he saw small curved bacteria in many patients with ulcers. This bacterium is now called Helicobacter pylori. I don't know if I'm saying that right, but good, close enough. He also saw the lining of the stomach in the ulcer patients was also swollen in the area near the bacteria. Barry Marshall was a doctor at the same hospital as Warren. The two men decided to work together to learn more about ulcers. Over the next few years, Marshall tried to show that H. pylori could cause, there's a picture of it, pretty cool, ulcers in healthy animals. By the way, this was done by a scanning electron microscope. Okay, you can't get pictures like that with a compound microscope. Over the next few years, Marshall tried to show that H. pylori could cause ulcers in healthy animals. He used young pigs for his experiment, but the pigs did not develop ulcers. People did not believe that his hypothesis that bacteria caused ulcers because he could not show it in animals. After years of no one believing him, Marshall decided to gather evidence by experimenting on himself. He used an endoscope, a long thin tube with a camera on the end, to take pictures of the inside of his stomach. He then drank a whole dish of H. pylori. After a few days, Marshall started vomiting. He was very tired, and his wife and colleagues noticed that his breath smelled really bad. After 10 days, he had taken another picture of his stomach and saw some of the bacteria that people with ulcers get. This provided convincing evidence that H. pylori was the cause of his sickness. There they are right there. Barry Marshall and J. Robin Warren accepting the Nobel Peace Prize in medicine. By the way, that takes a lot of guts to do. No pun intended there, but I don't think I'd have the courage to swallow a whole thing of H. pylori just to prove my theory. Kind of interesting. Over the next few years, Marshall and Warren showed that the newly discovered bacteria grew in the stomachs of almost all people with ulcers. They also showed that antibiotics cured these people. As a result of their work, ulcers became a curable disease. Marshall and Warren were awarded the Nobel Prize in Medicine in 2005. All right, boys and girls, that includes the reading. We have done the spontaneous generation. We have talked about that. So I'll go ahead and read it there. But this should be nothing new. The theory of spontaneous generation. Virchow stated that cells reproduce to create new cells. However, many scientists did not accept Virchow's ideas. They believed in spontaneous generation, the idea that living things grow from not living things. For example, someone who believes in spontaneous generation might think that plants grow from soil. If you wanted to grow a plant, you would need only soil, no seeds or plants or plant cuttings. After some time, a plant would spontaneously grow out of the soil. It took the experiments of many people to disprove the idea of spontaneous generation. In 1668, an Italian doctor named Francisco Redi set out to show that maggots grew from eggs laid by flies. Because maggots were often found in rotting meat, many people believed that they just appeared spontaneously. To test this hypothesis, he set up several flasks containing meat. Some of the flasks were open to the air, some were sealed completely, and some were covered with gauze. As he expected, maggots appeared only in the open flask, in which the flies could reach the meat and lay their eggs. In 1767, Italian priest Ozzano Spallanzini conducted experiments to disprove spontaneous generation. He tightly sealed bottles that contained liquid and then boiled them for more than 30 minutes. Nothing grew in the bottles. But because he had removed the air from the bottles using a vacuum, many scientists believed that Spallanzani proved only that spontaneous generation did not occur without air. It was not until nearly 100 years later, in 1859, that French chemist Louis Pasteur conducted a now famous experiment that convinced most people. The French Academy of Scientists had sponsored a contest for the best experiment to either prove or disprove spontaneous generation. Pasteur's winning experiment was a variation of the method used by Spallanzani. He put a mixture of yeast, sugar, and water in several glass flasks. He heated the necks of the flasks to bend them into the shape of an S. Air could enter the flask, but airborne microbes could not. Because of gravity, they would land somewhere along the neck of the flasks. Finally, he boiled the flask to kill any microbes that might already exist in the mixtures. As Pasteur had expected, no microbes grew in the flask. When Pasteur broke the neck of the flask and exposed it directly to air, microbes grew. Pasteur provided convincing evidence against the idea that living organisms come from non-living things. There's a picture of Pasteur's experiment. All right, boys and girls, have a great rest of your day.